Again, my name is Steve Darnell. I am a principal scientist here at DNASTAR, and I am the team lead of the structural biology group here. And we've been working hard over the past 6-8 months to bring the new features released in Lasergene 15 to the structural biology suite. As you may already know, DNASTAR has 3 main product areas that we focus in: molecular biology, genomics, and structural biology, and that's what my team and I work in. The central program of the structural biology suite is our molecular visualization and analysis program called Protean 3D. And it is a 3D structure viewer handling proteins, DNA, nucleic acid, structures, and sequences, but it does have a protein focus where you can visualize protein structure. You can analyze and perform bioinformatic methods along the sequence. The views are interactive so that you can interact with sequence analyses directly within the structure. We also have more structure analysis tools inside of Protean 3D as well, including the ability to create molecular and solvent accessible surfaces. You can calculate little region, or how much surface area a particular region is associated with. You can do structural alignments with Protean 3D. You can either choose to do full structural alignments, or you can make selections and align just those pieces as well. But today, or in addition to the structural biology suite, sorry about that. So we also have an evolving line of Nova products, which are tools that we've been developing over the past couple of years, designed to construct models, structure predictions, antibody structures, protein-protein complexes, and feed that data into Protean 3D for molecular visualization. So the 3 main areas and the 3 products that we currently have with structure prediction tools are NovaFold for doing structure prediction. It performs best with globular soluble proteins. NovaFold Antibody, a workflow that we've created for predicting how a heavy and a light chain of an antibody interact. And then NovaDock, which is a flexible protein-protein docking tool that explores molecular flexibility during a docking simulation. And again, all 3 of those get fed into Protean 3D. So we've licensed a number of critical pieces of intellectual property to help our some of these Nova tools along. NovaFold is powered by the iTaser algorithm from the University of Michigan. The principal investigator behind that work is Dr. Yang Zhang. NovaDock is based upon the SwarmDock algorithm from the Francis Crick Institute and formerly Cancer Research UK. The principal investigator behind that work is Dr. Paul Bates. And the NovaFold Antibody algorithm is actually a workflow that we have created here at DNASTAR, and that's what it is. It's a homology modeling workflow for quickly assembling antibody structures. So earlier this summer, we had expanded our cloud offering. Before, back in 2016 and before that, we had one product that was enabled on our cloud services, and that was NovaFold. But over the summer, we brought NovaFold Antibody and NovaDock onto the cloud as well. So since June, you've been able to predict or to initiate structure predictions for all 3 of these services directly through the DNASTAR website. The DNASTAR cloud itself is a secured platform where all aspects of the communication between your computer, the DNASTAR web server, and the Amazon cloud are secured. All the authentication is secured with standard SSL encryption technology. All communication is between your computers, the DNASTAR web service, and the Amazon cloud are fully encrypted. your data actually does not go directly through the DNASTAR web service. You upload your files directly from your computer directly to the cloud storage system. When you run a predicted calculation, you are not sharing virtual computers with anyone. We have isolated and dedicated computers for your prediction. So not only do you get on-demand computers assigned to you, they will be assigned strictly for you, and a set will be assigned for every job that you want to run to. So all of this is a fully automated system and managed, so that all you have to do is submit a job through one of our clients. The results are typically available sometimes, depending on the type of modeling, minutes. Otherwise, it could be a couple of hours. Structure predictions are downloaded very quickly, so you can download them from the DNASTAR web server, web app client, or as you'll see today, through Protean 3D itself. Now, this is, while we're talking today, I'm not going to go so much into a lot of the details of the individual tools this time around, but we have discussed these in prior webinars before too. If you're looking for a full overview of the NovaCloud services, how the software works, and how the results are visualized, we'll go over a little bit of it today. But for a much more detailed version, you could look at that first NovaCloud Services webinar. But we also have previous webinars about the web applications themselves on the DNASTAR website, and even more algorithmic information regarding NovaFold, NovaFold Antibody, and NovaDock. So that really kind of brings us to what we're here to talk about today, and that's what's new in Protean 3D. We have two dominant features that I'm going to be talking about today. So we have a new NovaCloud Services client directly within Protean 3D. Over the summer, to be able to do antibody and docking predictions, we were asking you to go to the DNASTAR website. Well, as of the Lasergene 15 release, which just came out last week, you can now perform all of these tasks directly within Protean 3D itself. You'll be able to do things in batch, unlike the website, which is designed to let you do individual predictions. You can submit a batch of predictions through Protean 3D today. And additionally, we'll talk about our new capabilities for doing site-directed mutation modeling as well. In the past, we've been requested about when we're going to bring this capability out, and it is now. And we'll talk a little bit more about the algorithms that are going on, just enough to let you get a feeling for where these modeling tools really are strongest. So with that, let's really get right into the software. So this is Protean 3D. What it typically looks like when you open it up for the first time. We're going to be looking at the NovaCloud Services client and workflows associated with it first. There are a number of places where you can actually get started. In our details panel, throughout your work in Protean 3D, typically when you make a selection in one of the other documents, you'll start to see more detailed information being presented here in the details view. And as we're working through this, you'll start to see some of that happen as well. But when we don't have anything open, we have this little Getting Started panel. You can start a new prediction from here, or through the File menu, or through the View menu. But let's get started by saying let's do a new NovaCloud prediction. This will be the login screen that you'll see when using NovaCloud. So NovaCloud is, again, as we were saying before, a cloud service. You need a DNASTAR account in order to use this particular system. If you don't already have a username and password for the DNASTAR website and cloud services, you can click on this link below for creating a new account, and that'll open up the DNASTAR website for you to help you start down that process. But I already do, so I'll just log in as myself. So there we go. When you log in, you will see that we have two views for the NovaCloud Services client. We have the Services views and we have a Predictions views. We'll start here with Services. This is a view that's simply designed to show you what sort of structure prediction workflows are available. And currently we have the three: NovaFold, NovaFold Antibody, and NovaDock. Each has a short little description describing briefly what it does, and it also describes what licenses you have available for using these services. If you don't currently own a license to any of them, you'll actually see a little note down here instead of where the licensing information is here. So that'll just be a place where, if you're ever curious to know how, what your license status is for any of these prediction services, you can go right here and see it. These are buttons that you can click in order to initiate any of the workflows. If you have questions along the way, you can come up here to this menu system and you can get directly into our help system, or you can, if you need to get to your account at DNASTAR's website, you can click on that, and that will take you to our account page. If you haven't already logged in, you'll be asked to, and then it'll take you right here for the ability to contact your representative, start to ask for help. It's also for as you'll learn if you read one of our other see one of our other webinars, we have the NovaCloud Services system through this login as well here. But we won't be focusing here. We will be focusing with Protean 3D again. So I'm just going to walk through the three workflows very briefly. When you click one of those three main workflow buttons, you will see a little wizard pop up for each one of these. And the general setup is that for each one, you'll first provide your input data. Typically, it's sequences or structures that you're using for modeling. You'll have the ability to set more advanced options, and then you can submit. So I already have a few jobs set up here. One of the things that I'm going to do is that I actually took for examples today, I took a structure from the PDB, and we're going to try to actually do some modeling of some of the individual components. Now, normally if you already have a structure, this is a lipoprotein bound to an antibody fragment. Now, when you, of course, have the structure, you know you already have the answer, and that's what you're looking for. But when you can use the NovaCloud services to help if you're interested in doing antibody engineering or prediction of B-cell epitopes or things along like that, you can use the NovaCloud system to model some of the data you would need in case you don't have structural data. So with NovaFold, you would model antigens. With NovaFold Antibody, you would model the antibody structures, and to predict conformational B-cell epitopes, you would use models from both of those tools and then dock them together using NovaDock. And of course, if you do have experimental structural data at any point, or external tool or structures generated from other tools, you can use those as well. So to start, though, we are going to, I'm going to open up just a FASTA file, or select a FASTA file of that lipoprotein. That file is nothing more than a simple FASTA file. I'll just open it up in a text editor quickly, and you'll see what it looks like with a little name. There's the sequence for it. We will pre-populate your job name with some of that information from the FASTA header. If you don't like it, you can just directly edit it right here in line. We'll also tell you a little bit about the sequence, specifically the the N and C termini regions of it, and the number of residues as well. If you don't have a file, or you prefer to enter it a little more manually, as if you were copying and pasting from a different file or from a website or something like that, you can directly enter them into this Enter panel. One thing that will happen is that you will see a little warning pop up if a full FASTA file had not been entered, or whether or not the sequence is a little too short or a little too long. So there will be some feedback along the way, and you'll start to see messages like this until you actually provide something valid for a FASTA file, but I won't use that. So we have a sequence loaded up, and we move on to the options. We have several modeling options here in NovaFold. We have the first option is this enhanced conformational search. This is a DNASTAR created tool that tries to perturb or morph selected templates through the modeling process, structural templates along normal modes of motion, trying to provide alternate conformational poses for during the modeling process. It's a way to include more structural diversity in modeling. Additionally, as you may know from other capabilities, we have in NovaFold, you have the ability to predict small molecule and ligand binding sites and protein function. You can control the number of models, the number of templates that are being selected, how much simulation effort will occur. You have the ability now in Protean 3D NovaFold client to add a PDB template either by name, or with a structure aligned to a sequence, or with your own PDB file that you can add to the template. I'm going to pass on those for the moment now. Additionally, you can add distance and contact restraints. You can look at the help for a little more details about this, but this is some sample examples that you can use to either define distances between particular atoms in two amino acids, or to state that two amino acids are in contact with one another. This little shadowy hint text will not be used in your prediction. Now, I am going to actually add a list of templates that I want to exclude. Because this is an actual PDB structure, this is the way you can actually say to don't use this particular model, 2YPV chain A. You can also define what sort of sequence identity that you want to exclude as well. So anything that is greater than 95% sequence identity to this template, we'd also remove as well. Or if you want to do it more globally, you can use this option down here to remove templates at a much coarser level at using sequence identity. When you have all inputs set up, you'll see all these values have been, or these status messages have been checked, and you can hit submit. You will see a little bit of status occur as we wait for the job to be submitted to the to Nova Cloud Services. The check mark here means it's done. You'd see a little red X if there was an error, with some error messaging to try to describe what went wrong. And if we look here, I'm jumping over to our predictions view. This is where you will actually go to monitor and manage and open and download result files. So this is the prediction that I just started just a minute ago, and that's what we're at. It's the elapsed time hasn't even been running for a minute, and we're at a starting state. I've already started some predictions this morning, and you'll see these are the sorts of messages that you will see as the prediction is running and what stages of the prediction process you're at. So NovaFold Antibody is very similar. You can provide a FASTA file that has multiple sequences, in this case a heavy and a light chain. Again, you can enter these manually as well. You can set some other options with regard to whether you're going to say that or require that the templates that are selected cover at least 75% of your structure. New to this release, you can actually have multiple H3 loop models. You can set that up between 1 and 5. You have the ability to control whether we use an ab initio style loop prediction or whether we use our more accurate fragment-based approach. And you can add PDB files to the library. You can include and exclude templates in a similar manner. And here, because this is a known structure, this is how you would actually say, don't use any of the protein chains from the original host PDB to YPV and use a little star for everything. And again, you can submit, and off you go. With NovaDock, the inputs are actually structure files, not sequence files, and you have to give a pair. You have to give a receptor, and you have to give a ligand as well. So in this case, we're going to set up another example. This is another fragment antibody that is has a structure that's bound to viral N9 neuraminidase. So in this case, people have asked me in the past, what should be the receptor, what should be the ligand? When I do antibody modeling, or in general, people kind of think, the larger molecule, the larger protein should be the receptor, and the smaller one should be the ligand, and that's fine in many cases. But if you want to take advantage of some known binding sites of a protein, it's actually better to make that be the receptor. So an antibody typically recognizes its antigen through the complementary determining region loops. And since we know by sequence analysis where those are, we can actually focus NovaDock to only search that region of docking. So I'm going to show you how to set that up. So again, here's our antibody fragment. I've already extracted the heavy and the light chains from that. The ligand file or receptor file is already extracted too. So defining this pair is all you need to do. Stepping through, you can find the number of normal modes of motion, which will be sampled for the receptor and the ligand. With antibodies, I like to use a higher number to have a little more fluctuation-styled capabilities. You can also ask for a larger number of docking simulations to perform by changing this value here. I've already constructed a list of the H3 loop residues, and this is how you define them here. You define the chain ID in the file, and then the numerical index associated with those residues. This is what's going to focus NovaDock onto just trying to sample the region around these residues. Moving forward and submit, and I'll just take a second, and there we go. So the predictions view is again where you'll see all this stuff. When jobs have encountered problems, you will actually see some error feedback like this. Additionally, when jobs you'll see the statuses that we've seen before, and when jobs are done, you'll see this little link pop up with a little bit of summary information. Again, the details of these are covered by other talks at the moment, but if you are interested in these, you can either look at those or contact us or me, and we can discuss these a little more in detail. But by clicking on any one of these, it will actually download the results for you and start to open up the file. From this view here, also you can stop and delete predictions that have already been run. Actually, wait for our NovaDock report to open, so there it is. So let's get on to the final feature that we're going to look at, and we are going to talk about mutating residues. So when you are doing site-directed mutations, substitution mutations, you need a starting structure. This example here is the pure E protein. It's actually enzyme. It's actually involved in purine biosynthesis, and there are already some structures with known where mutations have been created and crystallized. So there are experimental structures of the wild type and the mutated structures. So let's actually go and try modeling some of those mutations with the wild type structure that we have here. So I actually opened a file directly from my desktop. You can also open directly from the PDB. If you type in the same PDB ID, it'll download it and open it for you. But since we already have it here, let us work toward mutating two serine residues, serine 57, that occurs both in the first and second copies chains of this structure. So I have selected two residues, both serine 57 on chains A and B. You can either access mutate residues from here, a context menu in the sequence view, or from the structure menu itself. When you do that, we'll actually pre-populate any selected residues that you had into this table. You have the ability to add more manually by saying which chain in the structure that you've been selecting from, and the structure is listed here. I'm just going to type something quick in, and you can actually define the index. We'll tell you the residue, and you can pick a substitute. You could add this one directly. Or from when you have things selected here, you have to select residues from this list. And we're actually going to be doing an allele mutation since that's what's done in the PDB structure. If you prefer, you have the option to use 3-letter amino acid codes rather than 1 letter. And when you're done, all you do is you click the mutate button and it will run the process for you. We use some assumptions. We use a rigid protein backbone currently, and when we do the mutation, we're sampling a rotamer library and trying to repack both the mutated residue and all of the residues that are in contact with that residue. So we're trying to find the lowest energy packing arrangement from all the residues around and the substituted residue itself. It only takes a couple of seconds for a small handful of these, and you'll see a table with some results here, including the variant that we modeled, the calculated change in energy score. We use the D-fire potential to calculate these. You can also export this table for use later. And there we go. And it's a self-documenting file, so we tell you not only what the columns are, but a little description of what each one of these means as well. When we're looking at these energy scores, a negative score is a prediction that it's a more stabilizing mutation. A positive score is a prediction that it's a destabilizing mutation. And the magnitude is tries to describe the degree of scope between those. So those are energetic predictions of whether a point mutation is going to be stabilizing or not. So when you click the Finish button, what we actually do is we make a copy of that structure that we were modeling. It's been named slightly different than the others. All the other pieces are there, but what you will also find is that, one, we've made some new features in this document. In the Features view, the Variant tab, we will find the 2 mutations that we just made. Additionally, if you select these features in our Details view, you will see a little bit of information about the details of this and whether we noted that this was predicted to be stabilizing or not, the version of Protean 3D used, the energy score, and the potential that was being used. And these are they're presented as qualifier entries in a feature, so they are also listed here in the options bar as well. When you make a mutation, we actually they remain highlighted in the view when they start, and you'll notice here there's of these 2, I will actually select both of these, that we display the side chains for those for the new mutations as well. So the way to actually compare this against the original would be to display the side chains of both the wild type and the original, and then perform a structural alignment between the 2. If you feel, we actually go through, we actually try not to change the coordinate systems between the 2 things so that they stay superimposed. I'm actually going to open up an example where we've already done this analysis, visual analysis, where we went through and aligned all of the structures the wild type, the predicted mutations of those serine 57s, and also the observed mutated structures as well. So here's an example where I've already gone through, I've selected and rendered the side chains in a ball and stick mode for all three structures at the same position. So this was a larger residue to a smaller mutation. You have the ability to mutate to any other type of amino acid that you like as well. Because we are using a rigid backbone, though, that's trying to put larger residues into smaller locations, the structure is not going to perturb or deform around it, trying to let the backbone move. We do have plans to be bringing an additional degree of structural flexibility into this modeling process in our future releases. But I just wanted to let you know that so that you can think about how to use this tool appropriately and not to expect that we're going to be remodeling everything in the region. Right now we're doing side chain repacking around here too. But this is a great tool if you're interested in doing what-if scenarios, that you are curious into kind of sampling regions and seeing how side chain identities would affect the stability of the region or the protein. You can actually use this to quickly try to model mutated protein structures that were done so artificially to make them crystallize better and make them look more like the wild type if you're trying to use that construct to help guide some of your experiments. So those are our big two new features in Protean 3D. Mutations have been asked for a while, and I'm very glad that we've been able to bring those to you now. This will be kind of more of the tools that we're going to be developing over the next 6 months to a year as well as we move into more design-oriented workflows. But we are also want to keep these tools easy to use and fast to process when you want to use them interactively, and we'll kind of stop there for now. So, Sharon, I'll hand it back over to you, and we can answer any questions that anyone might have.
Great. Well, we've got a few questions from our attendees today, and the first question is, how does licensing work for the NovaCloud services?
So, with all the NovaCloud services, we offer two modes of licensing for purchase. You can either purchase term licensing. Typically, it's sold with annual licenses, but there are also more limited term licenses available if you want to speak with a salesman about that. We also offer per-use licensing as well if you feel that a term is not going to be appropriate for the types of projects that you're going to be working on. If you are concerned about using a cloud system, or you want more control over the entire process and are interested in bringing these tools and workflows into your own computational workflows, we also offer local versions for all three of them. They are currently command line tools, and they run on Linux Linux computing systems. But they are available for those who are interested in expanding their own pipeline or doing analysis in their own house.
Thank you. All right, the next question is, if I want to predict structures with variations in Protean 3D, should I use NovaFold or should I use the Mutate Residues feature?
Well, what I would recommend is, I think if it were me, I think I would start in Protean 3D most of the time. I would probably I'd model the wild type structure with NovaFold or bring an experimental one in, and then perform the mutations quickly and interactively in the view. If I were to suspect that maybe those mutations wouldn't really kind of fit in the area and that a larger degree of repacking or modeling would do, I would then actually consider using NovaFold for that, because NovaFold will have the ability to, it's a structure prediction product or workflow, and it does perturb local structure in an ab initio style technique through these larger types of simulations. So I think that would be better for grossly destabilizing simulations if you're trying to get a prediction of what some of the structural effects might be.
Thanks, Steve. And our last question for today is with regards to your mentioning that NovaFold antibody predictions often just take a few minutes, whereas NovaFold predictions can take a few hours. And our attendee wants to know why that happens.
Someone was looking at the predictions view, view closely, so thank you for catching that. In that view, actually, I will jump back to it quickly. There is a difference in time scale between these two approaches. Here are some completed predictions down here. These names with the fab and all, these were designed, just by me, just to say these are antibodies, but the little icons here show that these are, NovaFold Antibody or NovaFold and NovaDock. And you notice that, it took only 10, 20 minutes to do a antibody prediction. It's because NovaFold Antibody is a dedicated workflow for doing antibody homology modeling. We use protein threading to select what we believe are the most appropriate structural templates independently for both the heavy and the light chain. We assemble the two so that they're in complex with one another. We graft canonical CDR loop structures onto it, and then we use either ab initio modeling, or what I think is a little more accurate is a template-based, fragment-based approach to doing loop modeling for the H3 loop. And then we do an energy minimization. So rather than doing a lot of hard simulations, which is where the majority of NovaFold's time is spent, this is a more of a quicker homology modeling pipeline in its current form. We're thinking about doing more refinement capabilities to add on to it if people wanted a little more refinement effort placed beyond what the energy minimization and some of the rigid body optimizations are being done.